Okay, so more on chapter 6. So far we've looked at kind of how we study cells. We looked at the basic types of cells. We've talked about some of the um, pieces and components inside, like the cell membrane, what have you. So let's talk about the ER, Golgi, and lysosomes today. So this is part of what they call the endomembrane system. Think of this as like the highway or the transportation system for things inside the cell. This is how cells move raw materials, move food, move waste in and out of the cell. So there's a whole bunch of pieces to it that we'll talk about, but it's basically all membrane. So as we were going through the list of the parts of the cell that you need to know, which is pretty much every part and know their function, a common theme kept coming up is that these pieces of the cell or these components inside the cell are mostly made of phospholipids and proteins. Okay, this is why we spent some time earlier on talking about carbs, lipids, proteins, nucleic acids, because you're going to see this come up over and over again. And if you know how those basic things work, you can then make assumptions about how and judgments about how things that happen in larger scale organisms. So anyway, it's going to start with the nucleus or the nuclear envelope. It's going to be part of the ER, rough and smooth. The Golgi will play a piece in this, so lysosomes, vacuoles, and the plasma membrane. So obviously we talked a little bit about this in class. We know the ER really is it has two pieces, smooth and rough. It does account for the majority of the membranes found inside the cell. One of the things we talked about a little bit too was the fact that smooth ER it produces smooth ER produces. Sorry, something going on in the hall here. Okay, we're good. Smooth ER produces more membrane, and smooth ER produces hormones and steroids, where rough ER produces proteins and stores proteins. So kind of here's a quick diagram of it. So I'm going to zoom in a little bit. And one of the things you should notice is that, so here is the nucleus, and the nucleus has like this purple line here. All right, what that purple line is actually a space. So the membranes, the nuclear membrane or nuclear envelope has two sides. There's a membrane on one side and a membrane on the other. That's why they call it like an envelope. Right? It looks like an envelope you'd mail a letter. So that space is actually connected, if you look here, to the ER. So notice that the ER, it's got folded membrane, and there's a pocket or cavity in the middle. Kind of reminds me of like a pita pocket or a pita bread. So anyway, we can see these little pockets here. What happens is the ribosomes that are attached to these, the ribosomes produce proteins, and the proteins get dumped inside of this pocket here. All right, and they're stored there because they're probably harmful to the cell itself. Here you can see the nuclear pores. Okay, so once again, rough ER is rough because it's got those bound ribosomes to it. Here's a picture down here. This picture is from an electron microscope. It would be a scanning electron microscope picture. It's looking inside, and you can actually see the cavity there. You can see the ribosomes bound to it. You can see over here we have ER that's smooth. Once again, it's kind of cutting a cross section. So imagine this picture would be looking down from on high here, not necessarily on a 3D angle, but looking kind of straight down at it. Okay. Smooth ER. Smooth ER, once again, it's going to deal with making more phospholipids, steroids, oils, things like that. It's actually used also in detoxification as well. So there are enzymes in there that help with alcohol barbiturates. Barbiturates are like kind of hardcore drugs, kind of like OxyContin and things like that. Sometimes called opioids, our body knows those things have to be removed. So the smooth ER helps with that by creating these oils and lipids and steroids that will help actually deal with those things. So once again, rough ER is usually is more abundant in cells that produce proteins. So the term secrete or to ooze or to produce and send proteins out. Like for example, when you sweat, you secrete water, or when your nose runs, you secrete mucus. So these secretory proteins, or these are proteins that are produced inside the ER. So we'll go back here. So they're produced here in the ER. And then what happens is these proteins then are stored inside the ER, and the ER, a little piece of the ER will break off to produce a vacuole. So imagine that you have this, lack of a better expression, it's a little bubble of lipids that then that inside has enzymes. All right, these enzymes, all right, or proteins are going to be moved in and around the cell. So where this is going to go, it's going to leave the ER as a transport vesicle. But as it leaves there, so once again, a little piece of the ER is pinching off. Right inside that piece that pinched off, there's going to be some type of protein. Those proteins then will travel through the cell using kind of the cytoskeleton as kind of like a highway. And there are these little guys called motor proteins that help move this, which I'll actually post a video online for you to watch with that. It's really pretty neat. So that vacuole that then pops off is going to go to this place, the Golgi. So so imagine you have this highway system of tubes, the cytoskeleton, that are holding all of these cellular components in place, whether it be the Golgi or the mitochondria or the ER. And so it's a kind of like a suspension system of holding these pieces in place. As these small vacuoles break off the ER, the body doesn't know where to bring them. So what happens is you have these tubes, and these tubes that connect, let's say, from the ER to the Golgi, these microtubules. There are little motor proteins that look like little guys carrying packages that walk on these tubes. And the idea is they carry this vacuole, once again, this vesicle or vacuole, carries this to the Golgi. The Golgi then reorganizes it, so this vesicle vacuole kind of merges with the Golgi, sometimes called Golgi body, Golgi complex, Golgi apparatus. It's all the same thing. It merges with the Golgi because it's all just made of membrane. So in the Golgi, then there is a series of events that are going to happen where those proteins will be modified, will be repackaged, and they'll get a label, and then they get shipped out the other side of the Golgi, and they go to wherever they may need to go. Maybe they're going to be used inside the cell, maybe they're going to be used outside the cell. So depending on where they're going to go, the label that is slapped on the outside by the Golgi is what's going to determine where these things go. This is where you can imagine some problems happen. If you put the wrong label on it, it goes to the wrong place. If you don't put a label on it, maybe the cell reabsorbs it. So there's a lot of issues that can happen right here, which kind of, when we look at diseases that happen with people, we can kind of look and see what cellular component is actually causing it. It's pretty cool. For example, hair loss in men, I believe they've traced back to the Golgi not packaging the proteins that are made for hair follicles correctly, and so they never get shipped anywhere. So that's why you would get male pattern baldness. It's a gene issue, and that gene is carried on the X chromosome. And if you're a guy, it came from mom. Women can have male pattern baldness, but it's less likely. But we can kind of talk about it in class when we get to genetics. So you've heard this cis and trans, same and opposite. So we saw the word cis and the word trans, or those terms cis and trans, when we talked about isomers. So there's a cis side and a trans side. I don't think this is super important that you need to know what side receives or what side sends. Just know that these vesicles come in one side and they come out the other. The Golgi, to me, looks like a stack of pancakes when you look at it. So here's the Golgi apparatus right here. And these small circles here, these are all vesicles that are traveling to the Golgi. And what's going to happen is these vesicles, these little balloon-looking things here, are going to merge with the Golgi. Their contents will get stuck inside the Golgi. All right. So here's one. Once again, this one will come in. I guess it looks like coming in from this side. It'll merge with the Golgi, so the membrane that's around the vesicle will then become part of the Golgi. The contents will be stored in one of these cavities, or another term for it is a lumen. All right. And then there'll be a series of breakoffs. So let's say this part of the Golgi here, there may be a small vesicle that breaks off and merges with the next part of the Golgi, and then a small vesicle will break off and will merge to the next part of the Golgi. So the series, a cascade, or if you will, a series of vesicles being formed, and the vesicles that are finally come out the other side are the finished vesicles or vacuoles that will then go wherever they need to be. This is also how they make lysosomes. Peroxisomes are all made this way. Let's take a look here. So lysosomes, we talked about these in class a little bit. Lysosomes are primarily found, in only in animal cells. They are found in some plants. The exception to the plants would be plants that have to eat other things. Let's see, let's shrink this picture down a little bit. I think it's in the way. So, for example, if we're talking about the Venus flytrap, for example, the Venus flytrap is gonna have lysosomes because they have to get certain parts of their nutrition from other places because they're not able to pull nitrogen out of the soil. They need another source of nitrogen to produce amino acids. Those sources of nitrogens are basically bugs, so they take bugs and use them as a source of nitrogen. The other thing you see in here, in addition to lysosomes, we have like a mitochondria here. You can see by these little lines, it's the mitochondria. So these lysosomes, once again, they're digestive sacs, if you will. They're going to have enzymes in there that hydrolytic. So hydrolysis breaks things down. Hydrolytic enzymes are going to make hydrolysis, or the breakdown of proteins, lipids, carbohydrates, nucleic acids, happen faster. And they break them down into their monomers, which then can be either reassembled by whatever the organism is or used for energy. Certain cells will have more lysosomes than others. In this case, white blood cells will have the most, and the reason why is because their job is to gobble up other cells. You'd also probably see a lot of lysosomes in cells that line the digestive tract. Let's see. A couple things you need to know here with lysosomes. I think it's maybe the pH is important. So lysosomes work best at pH 5. So here's part of the issue. Your cells want to be around pH 7 or neutral. So this is about 100 times more acidic inside the lysosome than in the surrounding part of the cell. So one of the things that can happen is that we have this thing called autodigestion, or sometimes called autophagy, P-H-A-G-Y. Autodigestion or autophagy is a way for cells to be destroyed. So let's say that you have a cell that is toxic to the body. Your body can program cells to die, and the way they program them to die is they get a signal sent, and the lysosomes burst, and as those lysosomes burst inside of a cell, the digestive enzymes are released. The pH of 5 that's inside that lysosome is enough to stop the cell from functioning. There are some autoimmune diseases that this happens in, where the lysosomes actually cause cell death. We also have what they call programmed cell death when we're developing. I think I used the analogy in class. I think I asked who had webbed fingers and toes, and the answer is hopefully none of you. And the reason why is because during development, as you were growing limbs and fingers and things like that, some of those pieces that were there, and you have to think about your hands when they were formed, it was just a big ball. And as the bones were produced and those bones extended and grew, the skin was actually stretched between your fingers, so you looked like you had like a little webbed hand. The cells in those areas are programmed to die at certain times when they get hormonal signals from the brain. In the case of all of us, we have no webbed fingers and toes because the lysosomes in those areas actually cause those cells to die. All right, this picture is probably the most important part of this whole presentation. So here we have, and this is going to be for animal cells. Plant cells won't do this as much because they have a cell wall. So this becomes difficult of getting things in from the outside. So one of the ways we get things in from the outside is what they call phagocytosis. So there's two types of in-cytoses. So endo, E-N-D-O, cytosis is the process where things come into the cell. Exocytosis is where things go out of the cell. There are two types of endocytosis. One is phagocytosis. Phagocytosis means cell eating. It takes in large quantities of material. This material here would be called a phagocyte. The other things that cells can do in large quantities is take in large quantities of liquid. That's called pinocytosis, P-I-N-O cytosis. I have no way for you to remember those. I just think I think Pinot, there's a type of wine called Pinot, and I think that's liquid, and that's how I remember that. Phagocyte, I have no idea how to remember that one. You're on your own on that one. So anyway, so the food whatever the food is, the food particle is engulfed or swallowed by the membrane. So the membrane surrounds it, pinches off, and now you have this piece of food now that was too big to enter through the membrane is trapped in one of these little vacuoles here, or a vesicle. Either term is fine. Once it gets in, it gets in because on the outside of the membrane, which you can't see, there are receptors that grab that certain type of food, and then once the food is attached to that receptor, the cell membrane then pulls inside. The cytoskeleton helps make this actually happen. Once inside, the tag that was on the outside for the food is now flipped inside, so that your cell knows that, hey, this is a food vacuole, and that's where the lysosome comes in. The lysosome is then moved to the food vacuole. Okay, a lot of it's random in the sense that you'd have to have enough lysosomes to find the food vacuoles. It's a concentration issue, or the number of vacuoles you might have, or lysosomes you might have. And that lysosome will become one or merge, because it's just made of cell membrane, with the food vacuole, and then digestion takes place. So the enzymes that are in here have to be either, they could be general enzymes, but for the most part they're probably specialized enzymes that digest certain types of food. I'll use myself for example. If this was a hunk of cheese, not quite a hunk of cheese, but a molecule that was cheese molecule of some type, and it got into my cells, it gets into my cells, and what happens is I'm missing the lysosome that has the enzyme lactase to digest that. So I would get to the point where I wouldn't fully digest that. All right. Once digested, all right, the small molecules, so this large chunk of food is broken into smaller molecules, which then can diffuse out. All right, and the lysosome, or this package, can go back to the Golgi, get remodified to be a lysosome. Excuse me one sec. Back at it. So that's the kind of the process here. With this autophagy, if you look here, this diagram is supposed to be a broken cell component. This is probably a broken piece of the mitochondria. So in addition to kind of lysosomes taking food from the outside, we also have to clean up what's going on in the cell. So, think of the cell, they use a cell analogy of the cell being a city, and there's a lot of occupants in the city, and those occupants make trash. So you have to have a way of getting rid of that trash, so the lysosome can kind of work as a cleanup crew. It'll digest the whatever it is, in this case it's a mitochondria, will digest that mitochondria and produce little small macromolecules. So these are going to be, once again, carbohydrates, lipids, proteins, nucleic acids, which are made of sugars, amino acids, fatty acids, and glycerol, and nucleotides. Those will then get reintroduced to the cell, and they'll actually end up in the cytoplasm, so they can be reused. So cells are super good at, or do a great job at recycling the components they have inside. I hope this helps. This picture, like I said, this will be on the next test without a doubt. This is something you really need to understand how this process works. All right, have a good day.